All right, let's get started. This is my research proposal for the CWR proposal that we will do at the end of this class, and it is called Using Bulgaria Vesca F. semperflorens in Strawberry Fruit Ripening Research. And I just wanted to mention, if it's kind of hard to hear me, I am hoarse during this presentation, but I think I'll be able to get through it. Bulgaria vesca, which is the woodland strawberry, is a perennial plant that reproduces through clonal reproduction and through seeds. The species started to be cultivated in the 1300s in Europe. The woodland strawberry grows in forests in the North Hemisphere. It is the most common wild strawberry species. It can also be found in the Southern Hemisphere at higher altitudes. The woodland strawberry has a diploid genome, and it's used very much in the laboratory model for the cultivated strawberry, which I will talk about in just a minute. Fruit quality advancements are important in strawberry plant genetics. The production of fruit that is sweeter, longer lasting, and more beautiful is one of the main objectives of strawberry genetic advancement. Bulgaria vesca plays a huge role in understanding and locating specific genes that underlie volatile synthesis, anthocyanin accumulation for fruit color, and sweetness intensity. So let's talk a little bit about the diploid genome. So diploid is a term that refers to the presence of two complete sets of chromosomes in an organism cell, which each parent contributes a chromosome to each pair, just like humans. So diploid genotypes create more variability and fitness in the production than haploid genotypes, and they buffer better environmental change. As a consequence, if one wants to obtain good results for both average and peak fitness in a single population, one should choose a diploid population with an appropriate mutation rate. The strawberries that we cultivate today, Bulgaria ananassa, originate from two wild species growing in America, Virginian strawberries and the Chilean strawberries, which are from South America. These two Bulgaria species were introduced to Europe during the 18th century, and the hybridization into the new species garden strawberry occurred during the end of the 1700s. Commercial production in a small scale began in the 1800s. In addition to the woodland strawberries, the cultivated strawberries have two other closely related species in the Nordic wild flora. These included the creamy strawberries and the musk strawberries. The woodland strawberries are the most widespread, and they have the best fruit quality. However, if you are lucky to find a mature berry of a musk strawberry plant, they are very delicious. Creamy strawberries have a nice acidic flavor, but not the typical strawberry aroma. Creamy strawberries can sometimes ripen without becoming red. In vitro techniques are in place to propagate and conserve Bulgaria germplasm. This may be done in cold storage with liquid nitrogen as a meristem, shoot tips. So ex situ conservation includes seeds, in vitro cultures, shoot tips, dormant buds, and DNA. The demand for germplasms from strawberry breeders is always high. It is the utmost importance that germplasms with good traits are always available. The most common type of conservation is filled gene banks, glass houses, and net houses, where live plants are kept. Conventionally, conserving germplasm has proved to be high in input costs and labor costs. So the biotechnical tools such as cryopreservation, which is the liquid nitrogen, will help with these issues. Strawberry germplasm is being conserved in vitro in cryogene banks all around the world. And global conservation strategies have been discussed and started for wild strawberries like Fragaria vesca. Molecular geneticists know that there's advantage of working with the small-sized genome of Fragaria, and breeders plan to use the wild plant to expand the gene pool of the cultivated strawberry. So 27 countries and 2 gene bank networks have over 12,000 accessions of the strawberry in about 57 locations. Half of these accessions are advanced breeding lines of the Fragaria nassa, which is the cultivated strawberry that I talked about before. Support for improved health of strawberries and gene banks, training of gene bank staff, and coordination of web accessible databases of strawberry genetic resources are still needed and are being worked with a lot. The woodland strawberry can be used as a model organism for the perennial life cycle and the development of fleshy fruit. 68 microsatellite markers were tested in an accession of 298 Fragaria vesca plants. 56 of these were polymorphic and had an average of 4.46 alleles per locus. So they're this small oxygen upregulated RNA, which is the CWR gene, has been identified in Fragaria vesca. 66 of these genes identified in Fragaria vesca have been diversified expression patterns in vegetative tissues. There's further investigation into this type of gene, the CWR gene, functions in plant development, and fruit ripening will occur in the future. So let's talk about this Fragaria vesca F. semperflorens. This is the specific strawberry that has been chosen for this proposed study, and I will tell you why. So it is recognized as the best gourmet strawberry, excellent aroma and flavor. It's also called the Alpine Yellow Wonder. It does not produce runners. It's making for a great fit for borders, edgings, tight rows, and the gardens and containers, which is what it will be grown in for the study. Yellow fruit that won't attract birds, easy to pick, and once again, it has an amazing flavor. It's very cold tolerant, and the berries are about 1 inch long and a 1/2 inch wide, so they're way smaller than the cultivated strawberry. So Fragaria vesca may be used to quickly address questions of gene function relevant to these valuable crop species. Although some Fragaria vesca lines have been shown to be substantially homozygous, the homozygous populations exhibited a range of morphological and physiological variation. The genotype of Yellow Wonder is well characterized and it's sold commercially. An easy to grow standardized inbred diploid of the Fragaria line with a documented genotype. It is very available to all members of the research community and will facilitate comparisons of results among laboratories and provide the research community with a necessary tool for functionally testing the large amount of sequence data, and will be soon available for peach, apple, as well as strawberries. They're all in the rose family. Specifically, Fragaria vesca F. semperflorens, Yellow Wonder. They're easy to grow and can provide the research community with a tool for functionally testing the large amount of sequence for the strawberry. This line of highly inbred diplo strawberries are day neutral and runnerless, and the line produces sweet yellow fruit throughout the year under the protection of a greenhouse. F. vesca can be transformed with Acrobacterium tumefaciens, which is a soil bacterium that can be used to transfer a small segment of DNA into the plant. The success of this transformation makes the species a great candidate for RNAi in overexpression studies. So the picture is a microscopic view of the agrobacterium that will be injected into the plant. And I have continued to say how much the yellow wonder is sweet and is yellow and actually kind of tastes like pineapples. That was another interesting thing that I found out about this specific strawberry. So Bargeria vesca has a great potential to study the agriculturally important genetic information of the cultivated strawberry. So it's small, grows fast, has a large seed set, a small genome, and its sequence availability makes it a great model for developing new information about strawberry genetics. It has similar genomics to the cultivated strawberry and other cultivated fruit crops to be a very commonly used surrogate to test gene functions in the rose family species. The proposed studies outlined in this work wants to fulfill a specific use of the Emproid diploid species of semperflorens, which is the yellow wonder. Previous research has shown that the silencing of a ripening-related chalcone synthase, so CHS gene, can initiate new genetic studies on the ripening of fruit in strawberries and other species. The hope is to find new genetic material that may broaden the knowledge about strawberry fruit development and how it can be used to potentially advance ripening in cultivated species. Here are the research questions of this proposed study. What are the favorable characteristics in the genome of Bargeria vesca? How can Bargeria vesca studies be used to make advancements in the cultivated strawberry's ripening function? What can the inbred Bargeria vesca semperflorens, yellow wonder, show about the function and development of strawberry fruit? If the ripening-related chalcone synthase, the CHS gene that I talked about in the last slide, is silenced with the use of the Acrobacterium tumefaciens, then useful information about the ripening of the strawberry fruit will be found. An injection of Agrobacterium into not yet matured strawberry fruit of the yellow wonder strawberry should reduce the CHS gene activity and result in larger levels of the cinnamoyl glucose esters. The performance of this technique, along with metabolite profiling analysis, should uncover new discoveries in the genetic information in strawberry plant ripening, as well as other species in the rose family. So I have a picture of those other species, which I kind of already talked about. So the rose family includes apples and pears, peaches and cherries, and raspberries. The proposed study will occur at the University of Florida, since research for the yellow wonder strawberry plant has already been established in its horticulture science department. The strawberries will be grown in greenhouses in small pots. The yellow wonder is runnerless and can be maintained in pots and produce fruit year-round in a climate-controlled greenhouse. The strawberry plants will be grown by seed and will begin to flower after 8 weeks. The achenes of the first group can be sown after 4 weeks to start the next generation. 100 seeds will be planted per generation. Sodium halide lamps will be needed to give a day length of 12 hours. Next generation groups will be chosen based on the requirements of early flowering, robust appearance, number of achenes that enlarge after self-pollination, and a short number of days to harvestable fruit. In this study, yellow wonder will need to be cultivated with Agrobacterium tumefaciens that carries a green fluorescent protein reporter.
A colony of Acrobacterium will be grown in a medium with L. ripipicin, L. gemmatacin, and L. specimosicin in a thick solution. Final explants of the Agrobacterium will be blotted with a sterile filter paper in several different rounds. When clumps of shoots form, the entirety of the Agrobacterium will be placed in a hormone-free rooting media. The Agrobacterium strains carrying the specific IHP RNA gene will be injected into the receptacles of the maturing fruit of Yellow Wonder 14 days after pollination. So I have pictures here showing the Yellow Wonder strawberry plant once again. And then I have a picture of the specific lamp that will be needed to create a 12-hour day length, which is very crucial of getting the most fruiting to happen over a long period of time. And then I have another picture of the sterile filter paper, which is the circles next to the lamp, just to give you an idea of the different materials that will be needed. The sodium halide lamps, I looked them up just to see why it was so specific to have these types. They're just more efficient than other types of lights like LEDs. They just produce a different type of light and have better lifespan than LEDs. Callousing occurs in this proposed genetic study using Yellow Wonder and Acrobacterium, as previous research has shown. Shoot initiation, even after 9 weeks, will still not be very frequent. Organogenesis needs to occur in order for shoots to form. Shoot development and regeneration is best achieved with specific formulation called the ATSUCT or 2 promoter, and specifics on that promoter is in a work called Transgene Expression in Strawberries, driven by a heterologous phloem-specific promoter, and that work will be in the resources at the end of the slideshow if you want to see that entire citation. Also, since the proposed study is focusing on one line of strawberry plants, the Yellow Wonder, only genetic information will be researched and obtained from one very specific species. This study could be expanded to do injections in other strawberry species to see what might be discovered about fruit ripening and development. So I have pictures of other wild species. So the first one on the top left is the Virginia strawberry, which is also called wild strawberry. It is a ground-hugging plant rising from a fibrous perennial root system. It has hairy leaf petioles up to 6 inches long. Each bear a single trifoliate leaf. The hairy flower stalk gives rise to a loose cluster of small 5-petaled flowers, followed by tasty wild strawberries. Found in patches and fields and dry openings, this plant produces the finest, sweetest wild strawberry. The edible portion of the strawberry is actually the center portion of the flower, which enlarges greatly with maturity and is covered with the embedded, dried seed-like fruit. So the cultivated strawberries are hybrids developed from this native species and the South American one, which I'll talk about in just a second. The similar wood strawberry, Bargeria vesca, has seed-like fruit on the surface, not embedded, and sepals that point backwards. Then we have Bargeria chileinosis, which is the strawberry that originated from South America. It is on the top right. It's also called the beach strawberry. It's one of the 2 strawberry species used to create the modern garden strawberry, the cultivated strawberry. In spring to early summer, it blooms with beautiful white flowers, which are later replaced by unique red fruits. It has a dark green glossy leaves that turn red in the fall. It has a creeping habit and spreads at a moderate rate. Then the strawberry on the bottom has a darker appearance. This is the Fragaria moschata musk strawberry. It is astonishingly delicious and highly fragrant. These heirloom strawberries are from Italy, and they have fantastic flavor with hints of raspberry and pineapple, just like the Yellow Wonder. The sweet, soft fruits are almost round and smaller than more familiar strawberries, but they send out runners and make a very effective ground cover. I just wanted to point out these other wild strawberries that could be used in a study like this, where they can be injected with the agrobacterium just to see what else could be found. But Yellow Wonder, it has specifically already been tested through this injection, and proposing this study, I just knew that I needed to go further into that to see what exactly is it going to tell us. Is it going to give us more information about the ripening of the fruit? Now, if there was other studies using the other wild crop relatives, we could find other things like sweetness and the aroma and other things that the cultivated strawberry might need some advancements in. This proposed study can be performed within a 2-year span, mostly due because this strawberry species, the Yellow Wonder, is a fast grower, especially in a controlled environment. And the agrobacterium will need about 3 months to be ready for injection. Each generation of 100 Yellow Wonder strawberries will fully mature in 12 weeks, with a new generation producing fruit every 8 weeks. A total of 5 generations will be grown, which will take almost an entire year, with the injections occurring in the selected strawberry plants with the shortest flowering time, the largest number of enlarged achenes and shortest fruit ripening time. Results and discussion on the study will be completed 2 months after the research study and will then be published for the scientific community. So I just wanted to give some emphasis on the pictures that I put up here. So strawberry plants in this study, the Yellow Wonder strawberry, will be chosen depending on requirements on how much flowering will occur and how much fruiting will occur. Is it going to occur in abundance amount? And then it talks about those achenes, and they're going to have to pay attention to how many achenes will enlarge after self-pollination occurs. And so that bottom picture shows that when they enlarge, they're fertilized, and the unfertilized will be smaller. So that has to happen in order for those seeds to be ready to start the next generation. Collaboration with the Genetic Improvement of Fruits and Vegetables Laboratory, a USDA agricultural research service located at the Henry A. Wallace Beltsville Agricultural Center in Minnesota, will resume, as with the past studies on this specific research on the Yellow Wonder strawberry. So the reason why I have talked about the University of Florida and the Agricultural Research Center in Minnesota is because they both have already created a work that was studying the Yellow Wonder and researching and experimenting with the injections of the agrobacterium. So they already know something happens. With this proposed plan, is going to go deeper into it. We're going to find what it's going to show us, especially about the ripening of the strawberry plant. So the work is called An Inbred Line of the Diploid Strawberry Fragaria vesca F. semperflorens for Genomic and Molecular Genetic Studies in the Rose family. And that resource will also be on the bottom, which will be the next page when I get to my resources. It starts with Sloven, and it was also written by Smith and Fulta, 2009. I also wanted to mention that the picture in the greenhouse, that is researchers in the Agricultural Center at Minnesota. So that is actual performances going on there. I'm not sure what plant they're working with, but it's a very good example of what happens in Research Center. A lot of different plants are planted. There's a ton of plants planted in that example. So let's talk about this FAIR data. So FAIR stands for findability, accessibility, interoperability, and reusability. So this type of data will be created to share results with stakeholders. In plant research, phenotyping is usually shared directly with personal requests made by trusted relationships and technical publications. Social media is also a great incentive in broadcasting results, which can also inform the public. That is something that is what's making research faster. Like we have easier outlets of letting the public know, letting the scientific community know what we found in a specific research study. So along with the FAIR, just knowing that we have multiple outlets and easier ways to broadcast to people in a larger spectrum in this day and age because of social media, it's just a lot easier to do now than it was in the past. So up on the slide, I have a citation, and that is from Data Sharing in Plant Phenotyping Research, Perceptions, Practices, Enablers, Barriers, and Implications for Science Policy on Data Management. This will also be on my research slides, which are coming up. If you want to read more about that, that specific work is there. It's by Unchaku and Phillips, and it was written in 2022. So that is some very current information about FAIR data and how it's being implemented. So here are my resources. I've addressed some of them as I went through my slides. A lot about Fragaria vesca, and the specific SAUR gene is in the Kayak and Yulsenic citation. And I'm going to go on to the next page to tell you more about those. So that first citation on this page by Sloven, that is the one where the University of Florida and the Agricultural Research Center in Minnesota put together that work about Fragaria vesca F. semperflorens and its reaction, or its capability to be injected with the agrobacterium, and they have a lot of specifics on that. So if you are more interested in reading about the strawberry plant and specifically Yellow Wonder, why it was chosen and why it is so useful and resourceful to use it to learn more about strawberry plants. Also, the FAIR data citation that I talked about and the last slide is also on this page. It's right underneath slogan. If you want to read more about FAIR data, that's a very good resource because it was created just last year. Also, the citation for that specific promoter for shoots to come out for the specific study is in that last citation. So that is a very important aspect to make this study work because it will be very limited if shoot tips will not form. Thank you for listening. I hope you've learned more about the strawberry plant. I have. I've learned a lot about the just the different varieties and what's found in the wild and how that led to the cultivated strawberry. It is really neat to learn about the specific yellow wonder. I just never really thought about a yellow strawberry and how it actually has a really good taste and can be used in genetic research. So hopefully this all made sense. I tried to make it fun, and I hope everybody else had a good time making theirs. It would be very interesting to know if a further investigation of fruit ripening would occur with yellow wonder, but maybe some other thing will happen where they'll try to do fruit ripening tests on other wild varieties. And I don't think I mentioned this before, but like Semperflorens, the yellow wonder, it is very hybrid, very inbred from Fragaria vesca woodland strawberries. So it is cultivated. It is a strawberry that people plant in their backyards, but it is so closely related to Fragaria vesca. It's highly, highly inbred from the CWR relative Fragaria vesca. So Fragaria vesca in general, from the inbred lines of it, it is being used in research over and over and over again, and it just needs to get more specific now. We know that how well it's used because of its small genome and its fast growing. Now we need to go more specifically into what is it going to show us about the cultivated strawberry. How can it be used to make cultivated strawberries better?